Hello Year 11. Today we're going to do about genetic engineering. So we've already learned about selective breeding, and remember that is when you pick a trait you like. Say for example, you like a dog with floppy ears, so you find a dog with floppy ears and breed it with another one that has that same trait. You look at the offspring, you choose the babies that have got this trait, and then you breed them again. Eventually you've got lots of dogs with floppy ears that you can sell because they're really cute. The question on this slide, you can either do it now or you can come back to it later. It's a link back to what we've already done. So here it says you are going to breed a plant with purple flowers with a plant that has white flowers. So you've got one purple plant and one white plant. You breed them together. You only produce plants with purple flowers. What does this tell you? So you have to use your Punnett squares to do this. Okay. One of these traits will be dominant, one will be genetic, and you will be able to come up with a way that all of the flowers are purple. Okay, but that's not the important part for this lesson. This lesson we're going to look at genetic engineering. Now, I'm just going to show you this, and I'm going to explain it on a whiteboard. And we talked about this before. We talked about genetic engineering is used in insulin production. So if you know anyone that's got type 1 diabetes, I know some people have pets that have got type 1 diabetes, they need insulin shots. Now we used to extract this insulin from pigs, and that wasn't very good. You couldn't get a lot of insulin from a pig. Also, the insulin was not human insulin. Also, you are killing an animal to get something. We figured out a way to get a healthy human. When I say healthy, I mean they do not have type 1 diabetes and their pancreas can produce insulin. We take a body cell. We found the specific gene that controls insulin production and we cut it out. Okay, so here, we've cut the gene out. We then get a bacteria and insert that gene into the bacteria. So now this bacteria has a human insulin gene. Every time the bacteria then splits, if you're looking at the bottom here, every time that bacteria splits, it produces a tiny amount of insulin. And then we get all of that pure insulin off, and then we bottle it up, and then we send it away. Now, I've given you, this is quite the simple slide here for your notes. This is a bit more complicated. I would stick with the first slide, but if you are really confident and you really understand this gives you an application. It talks about insulin. Now, there is a word here called vector, which you need to know. A vector is just something that transmits something else, okay? So when we talk about diseases, a vector is something that carries that disease. And in genetic modification, a DNA vector is something that carries the DNA. Now, I'm going to get rid of this. Okay. Minimize that. I have made an ARC sheet for this. So please download this. You can, once you've downloaded it and saved it, you can do whatever you want with it. I've tried to put every single keyword. I've tried to simplify it as much as possible. Okay, and I've gone on to cloning and plants as well. So please use that. But for now, what I'm going to do is I'm going to get the whiteboard up. Here I've got a human cell that's got a gene in it. Now let's say specifically I'm looking for insulin. What I need to do is I need to find that insulin gene. Now this human cell here is healthy. It could be anything. It could be an arm cell, it could be a cheek cell, it could be a tongue cell. So I've picked a cell out of a human and it's healthy. What I'm then going to do is I'm going to use an enzyme. Okay, so I will actually type that. I'm going to use an enzyme, right? And I'm going to get this out. So first of all, I now get my DNA out, and I'm going to have my DNA squiggled in a straight line like this. Okay. So I've extracted my DNA. I've unraveled it. Now, because someone else has done it for me, I know specifically the gene that I want is there. That's what I want. Okay, I want this bit. So what I use is I get my enzyme, and they're like little scissors. Okay, my scissors, and they're going to come and they're going to cut along these lines here. So my enzyme cuts like this. Now I'm going to rub out these bits because now my enzyme has cut the bit that I want. I do not want anything else. So this is the good bit. Yeah. Now I've got the gene that I like. But that's not good enough. This gene's useless on its own. It needs to be in something. So if we go over to here, I'm going to go over to the right hand side. I'm going to find a bacteria. Now it's just a relatively harmless bacteria. Could be anything. We've just found the bacteria. Okay, that why is it doing this? Stop. There we go. Clicking the wrong button. We found the bacteria. Okay. And it's got its genetic code in here, okay, which we can't really do much to. But bacteria have these little circles inside them. This little circle is called a plasmid. Now, the little plasmid, okay, little plasmids will code for something specific. For example, maybe one bacteria evolved a way to survive in very cold environments. That specific thing will be put onto a plasmid and saved. Okay, because it's not part of the bacteria's main DNA, it's just sort of like a little evolution. What we do then, because we can't affect the bacteria's main DNA, right? So this is its DNA here. If we do that, we'll end up doing something to the bacteria, it could kill it. But this is separate, this plasmid. So we're going to take the plasmid out. Okay, plasmid is out. There it is. We then use our enzyme again. So our enzyme, we use a different one. So let's use a different enzyme. And what we do, remember this enzyme, I'm going to do a blue enzyme. Remember these are my scissors. There's my scissors. And I'm going to cut this open. I'm going to cut it open here. So it's still the same thing. I've just opened it up. So now my plasmid is cut open. I'll open it up like this. Now I'm an idiot and I've used them all in the same colour. So I'm going to recolor this purple over here. This human thing here is purple. Remember this bit. This was my human gene that I wanted. So what I've done so far, I've found a gene in here, I've took it out and I've cut it. So this is the gene that I want. I've then found the bacteria. I have removed the plasmid. I've cut the plasmid open. Now what I'm going to do is I'm going to get this gene and I'm going to put it in here like this. Now that's not good enough at the moment because it's just floating there. So then again, I get my enzyme. My enzyme there inserts this in, so it kind of glues it in. Yep. So my enzyme can be scissors or it can be glue. This time I've used it to glue this in. Now I have got this plasmid. And remember, this is human. This purple bit is human DNA. Okay? Now, the last step is I get another bacterium. It can be the same one, it can be a different one. So I'm going to draw my bacteria. There it is. Bacteria like this. It's got its DNA in up here, just squiggly, no nucleus. Okay, it might have a few plasmids in. But what I'm going to do is I'm going to get a scientist who's very, and they're going to inject this into here like that. So what I will have is I will have a plasmid that is mostly bacterial DNA, and then there will be a little bit of human DNA in here. Now this bacteria can produce insulin, and that's it. Okay, That is the whole process. It's quite complicated, but when you realize what you've got to do, it's quite simple. I need to get a bit of DNA out of here, this bit of DNA out of there, my specific DNA here, right, this little purple bit. I need to stick it into my bacteria. So I get my human DNA, I remove a little bit of the bacteria, I insert my human DNA there, and then I have to put this plasmid back inside to my bacteria. And there you have it. This bacteria can now produce insulin, and the bacteria doesn't even know it. Now, I'm going to link this. If my explanation wasn't good enough, because it's quite long, it might be worth watching this guy. All the notes that I've given you as well. I'd also like you to try and have a go at this exam question that's in the paper, in the folder. Now, have a go at it, but the mark scheme is at the bottom. So if you get stuck, okay, you can email me, or you can look at the mark scheme. Okay, and that's everything.